Hello guys, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my lecture. So today we are in lecture 3 of your excretion. So we're going to finish off excretion today. So we just have to talk about the mechanism of concentration of urine. Then we have to talk about the regulation of kidney function disorders. So with that we end this chapter. So ready? Yes? So shall we talk about excretion? So I hope all of your preparations are going on good, fine. So please continue your preparation. For all those who are taking up boards, yes, your curriculum has been announced, your curriculum has been released. So very soon we will discuss the CBSE curriculum. But as of now, let's concentrate on NEET, alright? Yes, Naveen, hello, a very happy good evening. Thank you for joining my lecture on time. So please do like, share and subscribe. So how is your preparation going on? Good? Yes, everything is good? Okay, so here we move forward and discuss about your excretion. So we were talking about all this. So we have to talk about the mechanism of concentration of filtrate, the regulation of kidney function, and what is MICTURITION. So here we go, a quick recap of the entire chapter, and then we'll finish off the chapter well before in time. So that's not an issue. So Monday we are starting off with sexual reproduction in flowering plants. So one more week we'll continue with syllabus wrap up, and in the month of August, yes, I will bring a revision session for those appearing for NEET 2021. Okay, yes, okay, so here we go. So we were talking about the human excretory system. So the human excretory system consists of what are the parts? So the human excretory system consists of a pair of kidney, then you have a pair of ureter, then you have urethra, and finally the urinary bladder, right? So we started off talking about the functions of your kidney, the structure of your kidney. Yes, Shimpi, hello, oh, very happy good evening. Yes, I'm good, thank you. How are you? How is Munna? All good? Yes? Okay. So we will talk about the structure of your kidney. So where is your kidney located? So it is bean shaped, it is reddish brown in color. So it is present between the last thoracic and the third lumbar vertebrae, right? So that is the location. So apart from that, the kidney is surrounded by a layer, by a tough capsule, right? So on the concave side of the kidney, we have a hilum. So it is a notch through which your ureters, your blood vessels and nerves enter the kidney. Yes, hi Leela, a very happy good evening. So thank you for joining my session. So please do like, share and subscribe. So Blessy, hello, hi. Yes, you're new to my session, so please do like and share. So thank you for joining my session. So all fine, okay. So I guess these days Munna is busy, yes, with her subjects, so she's not able to join our lectures, but say hi to Munna, alright? Okay, so the concave surface of the kidney has hilum, it's a notch, so that is where your blood vessels, limbs and the nerves enter. So inner to the hilum, you have funnel shaped structures in the kidney. So these funnel shaped structures are known as renal pelvis. So renal pelvis has projections which is known as your calyces, right? So how is the kidney structure? So let me draw the structure, right? So this is your structure. So here you have, so this is your renal pelvis, right? So here you have projections like this. So the projections are like this. So I drew it in a very easier way. So it is like this. So these are the projections, right? So these are calyces. So this is your renal pelvis. So this is your hilum. So where you have your ureter, you have your blood vessels, you have your renal artery, etc. So the outer layer is surrounded by a tough capsule, and the inner region of the kidney is divided into cortex and medulla, right? So the medullary region has conical shaped structures like this. So these are your medullary pyramids. So these are your medullary pyramids. So medullary pyramids open or project through these calyxes. So your the other one, the cortex region, moves through your medullary pyramids as columns of Bertini. So this columns of Bertini is your cortex region. All right. So you can see something like this in your kidney. So this is your columns of Bertini. Okay, so that is the cortical region. Yes, Dharni, a very happy good evening. So welcome to my session. So hi Anthony, yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So we are talking about the structure of kidney. All right. So the structural and functional unit of your kidney is nephrons. There are almost 1 million structure of nephrons. So the nephron has two parts. What are the two parts of nephron? So one is glomerulus, the other one is your renal tubules, right? So glomerulus is a tuft of capillaries. So tuft means a network of capillaries. So what brings blood inside the glomerulus? So that is your afferent arteriole. So the afferent arteriole brings blood to the glomerulus. So the afferent arteriole takes away blood. So if this is your glomerulus, the afferent arteriole takes away blood from the glomerulus, right? So this is the next part. So moving forward, so we also read about what is your Bowman's capsule. So here we have a capsule which houses your glomerulus. If this is your glomerulus, so this is your Bowman's capsule. So this together makes up your Malpighian tubule. So this Malpighian tubule is also known as renal corpuscles. Alright, so right below the renal corpuscles, what happens? You have a highly convoluted structure. So this is your PCT and then you have your loop of Henle. So then you have your DCT. So then it opens into the collecting duct like this. So this is the structure of your renal tubule, right? So this is your PCT. So this is your DCT. So this is loop of Henle. So this is your descending lymph and this is your ascending lymph, and this is, okay, so this is your collecting duct, right? So sometimes columns of Bertini, so columns of Bertini is when your cortical region, right, when it moves over the medullary pyramids as columns, so that is known as column of Bertini. So the inner surface of the kidney is divided into cortex and medulla, right? The cortical region, when it runs around the medullary pyramids as columns, that column is known as columns of Bertini. Alright, yes Shubham sir, a very happy good evening. So welcome to our session. So please do like, share and subscribe. Alright, so here we read something. So your Malpighian tubule, your PCT, your DCT, so and your loop of Henle, when everything lies in the cortex region, so such a nephron is known as cortical nephrons. So we have nearly 90% of cortical nephrons in your kidney, yes, and if your loop of Henle is present in the medullary region, so such a nephrons are known as juxtamedullary nephrons. Done? Yes. So this is almost 10% or only 5% that is present, not more than that. So then we read what is known as peritubular network. So the afferent tubules, the afferent arteriole, so it surrounds the network of renal tubules with what is known as peritubular capillaries. So these peritubular capillaries form a minute U-shaped vessel called as vasa recta, which is parallel to your loop of Henle, right? Yes Raja, very happy good evening. So welcome to my session. So please do like, share and subscribe. Hi Mercy Mary, a very happy good evening. Yes, welcome to my lecture. So this is one half. Then we went on to talk about the mechanism of urine formation. So what are the three steps in urine formation? The first one is glomerular filtration. So glomerular filtration is also known as ultrafiltration, because the purification is so very perfect that even plasma proteins are not allowed, right? So the next step is your reabsorption, and the third one is your tubular secretion. So here you need to remember certain points. So in glomerular filtration, so 3 layers are present, right? So one is the epithelium of the Bowman's capsule, which is called as the podocyte, which has the filtration slits. Then you have the endothelium of the glomerulus. Then you have the basement membrane between the two. So the filtrate that is present or that is filtered by the glomerulus is known as the glomerular filtration rate. So this glomerular filtration rate, right? So this is 125 ml per minute, or it is 180 liters per day. So this is the rate. Suppose if this falls down, if this flow falls down, if the glomerular filtration rate falls down, then you have the activation of renin-angiotensin mechanism. Yes, you remember what is renin-angiotensin mechanism? We'll talk further about it in the regulation of kidney function. As of now, hold on. So what is reabsorption? You reabsorb from the tubules, right? So that is reabsorption. So you should remember one main difference between reabsorption and secretion. So where it gets reabsorbed? From where to where? So tubular reabsorption is from the filtrate to the blood in the peritubular capillaries. So this is where it is getting reabsorbed. So you're reabsorbing what is there in the filtrate, right? So this filtrate is there in the renal tubule. So this you should remember, alright? The next one is secretion. So tubular secretion. So from where to where it is secreted? So from the blood of peritubular capillaries into the filtrate in the renal tubule, secretion happens, alright? Yes, Kalpita, a very happy good evening. Yes, thank you for joining my lecture. Okay, so this is secretion. So this is about urine formation. So then we went on to talk about the function of your tubules. So what is the first tubule? That is PCT. So proximal convoluted tubule. So maximum all electrolytes, nutrients are reabsorbed here. So it helps in reabsorption of essential nutrients. It also helps in the process of tubular secretion. The next one is loop of Henle. So loop of Henle has descending lymph first. So descending lymph is permeable to water, but it's impermeable to electrolytes. So that's what happens. Water moves out of the descending lymph. So thus the filtrate is concentrated here. So here the filtrate is concentrated. So this is the conclusion at the end of descending lymph. So how about ascending lymph? So ascending lymph is vice versa. It is permeable to electrolytes and it is impermeable to water. So water stays inside, so your electrolytes move out. So thus the filtrate here is much diluted. So then we went on to talk about your collecting duct. Your collecting duct does conditional reabsorption. Your distal convoluted tubule does conditional reabsorption of your Na plus and electrolytes, to some extent even urea. So it also reabsorbs water. It is also involved in the process of secretion. So collecting duct helps in reabsorption of water as well as secretion and reabsorption of urea. So this is the function of your ducts. So one more thing I want to tell you. So before we go into this, so what is metanephric and what is mesonephric kidney? So that is the doubt which many of the students get. So what is mesonephric kidney? So mesonephric kidney has nephrons, has even Bowman's capsule, but it is not well developed, alright. So example, it is present in birds and reptiles. So the next one is metanephric. So metanephric kidney is one which has nephrons, which has Bowman's capsule, which has loop of Henle. So it is seen in humans or higher mammals. So this is the difference between metanephric and mesonephric. Pronephric has no well developed nephrons. Alright. So now we are talking about the most important concept in your excretion, that is mechanism of concentration of filtrate. So sometimes you do not drink enough water. Right. So we forget in our busy schedule, we forget to drink enough water. So what we do? Nothing, right? We don't drink water and we realize, okay, we haven't drunk enough water. But for your body, it is too late. So it has to conserve water at some point. Otherwise, your body will run out of water. The major chunk of your body is made of water. So the body needs to be supplemented with water, right? So this kidney has to conserve the little water that it has. So how is it going to conserve water? Only by reabsorbing water and concentrating your urine. So this mechanism of concentration of filtrate is very, very important. That is the way by which your kidney conserves water. Alright? Yes? Fine. So now moving forward, how does it do? How does the kidney concentrate the filtrate? How does it make it more concentrated and remove water? So this is done by two main parts of your kidney. One is loop of Henle, the other one is vasa recta. Okay, so to concentrate your filtrate, you need two gradients. Gradient is difference in concentration. First, you need a gradient of NaCl. Next, you need a gradient of urea. Alright, so for that, you should understand that your vasa recta and your Henle's loop are opposite in flow. Right? So this is your vasa recta. So according to your vasa recta, so this is the descending lymph and this is the ascending lymph, right? But in our loop of Henle, it's the other way around. So this is the descending lymph and this is the ascending lymph, and these two are placed side by side, right? So since they have opposite flow of the content inside them, so this is known as counter current mechanism. So this is known as counter current mechanism. Fine? Yes? Okay. So now how does this process happens? So we need two gradients. One is gradient for NaCl, the other one is gradient for urea. So how does this happen? So before that, you should understand we have cortex region, we have medullary region. So in the medullary region, you have more concentration of ions. That is, you have increased osmolarity. Osmolarity is nothing but the concentration of your ions, right? So inside your medullary region, you have more concentration of ions. Thus, the osmolarity is 1200 milliosmolarity per liter. So the osmolarity inside your medulla is higher compared to your cortex. Here it is only 300, here it is 1200. So more ions are concentrated in the medullary region. Fine? Yes, Swami, a very, very happy good evening. Thank you for joining my lecture. So please repeat it. Okay. So let me draw the structure. So we have your PCT. So from PCT we have your loop of Henle. So please bear with my diagrams. Okay. So this is my loop of Henle. Right. So this is my descending lymph. This is my ascending lymph. So now my vasa recta is like this. So in vasa recta, this is the descending lymph, this is the ascending lymph. So this is vasa recta, right? The flow inside both of them is in the opposite direction, right? This is ascending, this is descending. So they have counter current mechanism. That is opposite in flow. So this is point number 1. For concentrating urine, you need two gradients. So gradient is difference in concentration. So this gradient is seen in case of NaCl or urea. Fine? Okay. So, you should also remember the osmolarity concentration. So, osmolarity is greater in the medullary region compared to the cortex. So, in the medulla, the concentration of ions is 1200 milliosmolarity per liter. Okay. So, in the cortex region, the concentration of osmolarity is only 300 milliosmolarities per liter. So, here the concentration of ions is more, right Shimpi? So, this is what? This is your medulla and this is your cortex. So, here my value is 300. So, here my value is 1200. Right? Now, how is this going to happen? So, first we are going to talk about the gradient of NaCl. So, NaCl, we know the ascending limb of the loop of Henle is permeable to electrolytes. So, your NaCl comes out from here because this ascending limb of Henle is permeable to electrolytes. So, NaCl comes out, right? Yes? Okay, so that is different. Concurrent, okay, so just leave off. Now remember only counter current. So that is in detail. I am going a bit fast. Yes, there is one more thing known as what is concurrent. So this is counter current is two different directions. Concurrent is almost the same direction but in different concentration. Okay, I will tell you. So this is counter current. That is when you have opposite flows. Yes. So, there is another term that is known as concurrent. So, concurrent is I have one tube. So, it starts from 0 and it reaches 100. There is another tube which starts from 100 and it reaches to 0. This is concurrent. The concentration differs but the direction of flow is same. Okay, Antonia B? Yes, it's good that you recollect things much quicker. Fine. So, here your NaCl comes out of the ascending limb because this is permeable to your electrolytes. So, when it comes out of the ascending lymph, it is collected by the vasa recta's descending lymph. So, it enters inside the descending lymph of vasa recta and it once again comes into the mediastinum region. It once again comes into the medullary region. So, once again inside the tube your ions are getting concentrated. So, in the medulla the osmotic concentration is pretty high, right? The concentration of salt is pretty high. So, thus water moves out. So, here when you have, so ideally what happened? Water moves out because you have more amount of ions, right? Okay. So, this is about your NaCl. Now, what about your urea? So, you have your collecting duct here. So, the collecting duct has urea. So, urea comes out of the collecting duct and once again it moves into the descending lymph of vasa recta and it reaches the mediastinum. So, thus water moves out by the result of osmosis. So, osmosis is movement of water from higher concentration to lower concentration, right? So, if everything inside the tube moves out, so here NaCl is moving out, here urea is moving out. So, inside the tubes you have high concentration of water. So, now water is getting moved out. So, here also I have some amount of water, here also I have water, here also I have water. So, now if your ions come out, it is filled with water, right? So, now by the process of osmosis, your water moves out and now the filtrate is getting concentrated. So, at the end, the filtrate becomes hypertonic to blood, increased in concentration of ions to blood. So, thus this is how your kidney conserves water. Is that clear? You need to remember there are two concentration gradients, one for NaCl, one for urea. Is this clear, guys? So, the mechanism of concentration of urine is clear to everyone? No doubts? Yes? Can we move forward to the next part of the story? Yes? Can I? So, here we go. So, next we are going to talk about the regulation of kidney feedback. So, who regulates the kidney feedback? So, here we go. So here we talk about the regulation of kidney feedback. So it is regulation of kidney function. So you have three types of regulation. So first regulation is by your ADH or your vasopressin. The second one is your renin-angiotensin mechanism. And the third one is your ANF mechanism. So all of these regulate your kidney function. Done? So the first one, let's talk about ADH. So whenever there is a change in body fluids, change in the volume or concentration of your body fluid, so you have certain receptors in your body which is called as osmoreceptors. So these receptors get activated. So if these receptors are activated, they initiate or activate your hypothalamus. So hypothalamus in turn activates your posterior pituitary, that is your neurohypophysis, to release what is known as ADH or vasopressin. So ADH is antidiuretic hormone, so it reabsorbs water. It causes conditional reabsorption of water. From the DCT. So this is one type of mechanism. Alright. So this is the first regulation. So this is ADH or vasopressin. So now moving on to the second type, if there is a decrease in the GFR rate, if there is a decrease in the GFR pressure, so then what happens? So in the junction between your afferent arteriole and your DCT, so we have what is known as juxtaglomerular apparatus. So these cells get activated and they produce an enzyme known as renin. So this renin converts angiotensinogen. So tensinogen is the inactive form into angiotensin. So angiotensin, what it does? Before that, here your pressure is less and you have increased volume. So pressure and volume are inversely proportional. I have told you n number of times, right? So here the pressure is less, so the volume of the blood vessel is greater. So here your angiotensin constricts the blood vessel. It constricts the blood vessel. So if you decrease the volume, automatically pressure increases. So you have normal GFR flow rate. So this is renin-angiotensin mechanism. So number 3, what is ANF? ANF is natriuretic factor. So once again, when the blood pressure in your heart is very high, okay, so when the pressure inside your atria is very high, so the heart walls release ANF. So once again, if the pressure is high, the volume is low, right? So ANF increases the volume and decreases the pressure. So this is a blood vessel dilator. This is vasodilator, right? And this is vasoconstrictor. So this is how your kidney functions are regulated. Alright, is this clear to everyone? Yes, any doubts? Is this clear? No doubts? Can I move forward? Yes? Done? Okay, so after this we move into a process what is known as micturition. So micturition is the process of release of urine. So how do you release urine? So it is a neural mechanism and it is called as micturition reflex. So how does this happen? So you have what is known as stretch receptors on your urinary bladder. So urine is stored in your urinary bladder. right? So once these bladders are full, they send signal to your brain. So who in the urinary bladder sends signals? So you have what is known as stretch receptors. So these stretch receptors, they send signal to your brain saying that the urinary bladder is full and we need to release urine. So these stretch receptors send signal to the brain, so which in turn causes the voluntary action of releasing urine, that is micturition. Alright, so what is the pH of urine? Any idea? The pH of urine is slightly acidic and it has a yellowish odor. It is yellowish in color. So this yellowish in color is due to a pigment called as urochrome. So this urochrome is formed by the breakdown of hemoglobin in your body. So your RBC cells have a lifespan of 120 days, right? After which it has to be degraded. So when you degrade RBC, that gives a particular pigment called as urochrome. So that gives the color to your urine. It is slightly acidic and it has a specific odor. So after you pass urine, it stinks. That is because the bacterial decomposition is taking place, which converts urea to ammonia. Alright, so this is about your urine. Fine. So moving forward, so we are done with the regulation. So we are into the last part of the topic, that is disorders of excretory system. But before that, what are the other organs which are involved in excretion? So the other organs which are involved in excretion, first one includes your lungs. So lungs eliminates carbon dioxide and to some extent it also eliminates your urea and your nitrogenous waste. Okay. Then you have liver. So liver eliminates your bile, your steroids, your drugs, etc. And number 3, you have your skin, right? So skin has your sweat glands and it also has your sebaceous glands. So sebaceous glands secrete oil and wax. So it has hydrocarbons, steroids, etc. and this produces sweat which has your NaCl, etc. So these are the other organs which are increased in your excretion. Yes, lungs excrete carbon dioxide, right? They oxygenate your blood. So that is where the process of diffusion happens. Right, you are right, Leela. Very good, fantastic. So we move forward with what is known as excretory system. So disorders of excretory system. So first is uremia. So uremia is when your kidney malfunctions. It leads to the accumulation of urea in blood. Okay. So what is the function? So what is the remedy? So the remedy is hemodialysis. You want me to repeat the pressure terms? Yes, I will repeat after finishing the disorders. Okay, Anthony Abhi. So malfunctioning of the kidney is uremia. So you can either treat, but if you have prolonged malfunction, then it leads to kidney transplant or dialysis. So you have what is known as hemodialysis. So dialysis is a process by which your body cannot excrete, but you depend on a particular machine. So they take blood from your renal artery. Okay, so they take blood from your artery, not renal artery. Then they pass it into a dialysing machine which has a dialysing fluid. So it has some concentration similar to your plasma, such that the concentration gradient is created. So there is a membrane inside your dialyzer, so which causes the waste substance to be eliminated, and once purified, your blood is passed via the vein inside your body. Okay, so that is hemodialysis. Then acute renal failures. So it's definitely a complete failure and it's a prolonged failure. You cannot continue dialysis for years together. You can only do dialysis for months, because a single time of dialysis takes 5 to 7 hours. You have to sit in a particular place. That is not possible, right? So acute renal failures, the only replacement is transplantation. Then renal calculi, so this is your kidney stones, so insoluble mass of crystallized salts. Then you have glomerulonephritis, so this is your inflammation of your glomerulus. Alright, yes. Yes, anticoagulant heparin is added because if you take the blood from your artery, so it has a tendency to clot, but we don't want it to clot, so we had anticoagulant factors, that is heparin. So once again, if you pass the blood inside your body, you remove the anticoagulant and then put it inside, okay. So glomerulus, so these are the disorders of your circulatory, of your excretory system, fine. So now Antony wants me to repeat certain pressure terms. So you have what is known as renal plasma flow. So renal plasma flow is the blood entering the kidney. So about 1/5 of the blood from the heart enters the kidney, right? So that is renal plasma flow. So how do you know if your glomerulus has done proper filtration? So what are the driving factors? So the effective filtration rate. So who decides this? So this is decided by three things. One is glomerular hydrostatic pressure. So this is GHP. The second one is colloidal osmotic pressure. So this is COP, right? So this is the second one. So what is the third type of pressure that I taught you? So the next one is capsular hydrostatic pressure. So what are these? So glomerular hydrostatic pressure is the pressure inside the glomerular capillaries. So it is due to the difference in diameter of your afferent and your efferent arteriole. So this is the pressure within your glomerulus. The average value is 60 to 70 mmHg. So this is the pressure within your glomerulus, right? And this is due to the difference in the diameter of your afferent and your efferent arteriole. Fine? Okay, so then you have colloidal osmotic pressure. So the colloidal osmotic pressure is the pressure that is present in your blood capillaries, in your normal blood capillaries, because your plasma proteins are not filtered. So there is some type of pressure here, that is colloidal osmotic pressure, right? So what is the value of colloidal osmotic pressure here? Yes? So why? What is your problem? Why are you not understanding? Yes? So this is around say 20 to 30 mmHg. Yes, Avinash, what is your doubt? And the last one is capsular hydrostatic pressure. So now your filtrate is filtered and it comes down the renal tubule, right? So there is some pressure within the filtrate as well. So that is your capsular hydrostatic pressure. So here it is the pressure within the filtrate that comes out of your glomerulus. So here the value is around 10 to 18 mmHg. So now how do you calculate effective filtration pressure? So effective filtration pressure is equal to GHP minus your COP plus your CHP. So what is it? It is 60 minus you have 32 plus 18. So we take the average values here. So this is around 50, right? If I am right, yes, this is 50. So the net effective pressure is 10 mmHg. All right, Anthony Abhi, did you understand? Yes, Shamita, a very, very happy good evening. So I hope you're doing well, but you're towards the end of my session. I hope your preparations are going on better. So please do like the session and thank you for joining my class today. Yes? Yes, now it's clear, Anthony. So this is the formula. So this is the pressure inside the glomerular capillaries. This is the pressure inside the blood and this is the pressure inside the filtrate. Done? Any more doubts? So with this we are done with excretion. So one more week okay, so this is renal plasma flow. The blood that is entering the kidney from the heart is your RPF, renal plasma flow. Right? Okay, so one more week I will move on with syllabus wrap-up series. After that, we will start something new for those aspiring for NEET 2021, and we are very soon gonna start board classes as well, because the board has new pattern. So very soon we'll be starting board classes as well. So in the next week, we are gonna finish 2 chapters and 1 mentee. So we have to take a syllabus wrap-up mentee, right? So then we'll finish off locomotion and movement. We'll also finish off sexual reproduction and flowering plant. So this is the plan for next week, and after that we have a new plan that I will let you know tentatively. Done? So please prepare well. Any doubts, please get back to me on Telegram. So I'm always ready to help you. So if you have not understood, so please watch my lectures once again, go through them, solve a lot of questions, recollect the concept. That is very, very important. Okay? So hard work, determination, persistence is very much important at this time. Yes, we are counting days to go there. So yes, I will let you know when the mentee is, when the mentee topic is. So mentee topic will be your genetics and evolution. Alright. So are there any board students here who are going to write 12th standard board exam this year? That is in 2022, are you going to appear for 12th board exam? Any students of that category? Yes. So please be ready. We are going to begin board classes as well. So we are going to start in a new pattern. Yes. Okay, so you are a 12th, you are a CBSE student. All right. Okay, guys. So thank you so much. So this is me wishing you more and more. So please prepare well, and any doubts, get back. So have a great weekend. I will meet you on Monday. So Monday at 5 p.m. we are talking about sexual reproduction and flowering plant. All right. No, today no mentee. So next week we'll have a mentee. I will let you know what time, what date, and what syllabus. Okay. Chalo. So have a nice weekend. Bye bye and take care. Okay.